Hi, my name is Laney Widener, and I'm a second-year master's student in the Plant Biology and Conservation program, a unique program with Northwestern University and the Chicago Botanic Garden. Tonight, let's talk biodiversity. There is an amazing array of organisms on our planet today. Did you know there is 8.7 million known species on the planet? Species of animals, species of plants, species of fungi, and that doesn't even include the little guys like bacteria. We as humans have devised a way to categorize or organize this life on our planet, a science we call taxonomy. And taxonomy is not just an academic endeavor. It is a fundamental task for biologists. We use taxonomy to pollinate our crops, to manage pests, to improve human, animal, and plant health, to combat climate change, and to help conserve our environment. From our beginnings as a species, we needed to categorize life on our planet. We needed to know which plants were safe to eat and which plants were poisonous. We also needed to know which animals were harmless, like a house cat, and which animals were dangerous, like a wild wolf. In the 1920s, a mealybug was introduced to the coffee crops in Kenya, and this mealybug wreaked havoc on the coffee plants. It caused about 10% of crop loss every year, and twice we tried to identify the species, and twice we failed. And this consequence caused coffee crop loss and coffee profit loss. And it caused wasting time and resources with the coffee farmers trying to control this mealybug. Then we finally did a taxonomic study of mealybugs and found it was a completely new species of mealybug. With this information, we were finally able to control this pest. Taxonomy is not an easy task. Just like it'd be tricky to identify different mealybugs, it's not so easy with plants either. In plant biology, oftentimes plants are first identified as different species by their physical features, their flowers, and their growth form. But this can be deceiving. Here are 3 species of plants in a group I study called paintbrushes that look very similar to one another and are a little hard to tell apart. Geography can help us in a way because where they're located in the US can kind of help us tell what species are where. However, in California, the 2 out there have overlapping ranges in habitat and they coexist together. So that brings back the problem of trying to identify these species again. Today, we have amazing technology to really delve into the differences, the genetic differences between these plant species. Think of genetics like a fingerprint. Each plant species has a unique fingerprint pattern associated with them, and like the fingerprint, they have a unique genetic ATCG code also associated with them. From the genetic codes, we can start to compare these species and tell them apart as separate species. And in comparing them, we can look at similarities and differences between the species to start to understand what a species is. The 2-barred flasher butterfly was once considered a single species of butterfly from the appearance of the adult butterflies. In 2004, we did a genetic study of these butterflies, comparing the adult butterflies to one another, and found there were 10 different species of butterfly. And these cryptic or hidden mysterious species show us that it's so important to include this genetic codes in comparing the different species, because we may not even know what biodiversity is out there if we have a lot more cryptic species. In my research, I look at 6 different species of wildflowers along the coast of California, and I'm using both physical features and genetic features of these plants to look at and compare them as species. And I can use that to build this sort of family tree of relationships, of how they're related to one another as a species. I can also look at speciation, how species are formed, how these plant species are formed, and how they're maintained or stay separate plant species. California is one of the biodiversity hotspots of the world, meaning there's a huge amount of biodiversity in a very small area. And this may be partly due to the different habitats found there, like mountains and forests and deserts. And when we zoom into one like the coastal, where my wildflowers are, we can really start to look at the questions of what kind of diversity is within an ecosystem and how this diversity formed. And we can look at closely related species, like in my plant species, to really ask, what is a species? And knowing what a species is very important to plant conservation. The Tiburon paintbrush is a federally listed as endangered plant species, meaning it's threatened with extinction. And the work I'm doing with plant taxonomy and genetics, I am providing data and information to Creekside Science, a restoration and land management organization who is working to restore these plants and their habitat so that we don't lose them. The Tiburon paintbrush is not the only plant species endangered in the US today. There are 877 federally endangered plant species in the US today. And if we zoom in to state level, there are 332 state endangered and federally listed plant species in the state of Illinois. My work with plant taxonomy and plant genetics can be applied to other plant species like these Illinois natives. And applying this to other species, we can start to conserve endangered plant species through science-based habitat restoration and land management. And from there, we can help to prevent the loss of the amazing biodiversity we have on our planet today. Thank you.